Welcome to This Explainer. Today, we're diving into the incredibly vibrant and honestly fascinating world of genetic and pediatric diseases. We're going to take a journey of scale together. We'll start by looking at the global impacts of these conditions, and then we're going to zoom all the way down, past the organs, past the tissues, straight into the nucleus of a single human cell. It's an absolutely incredible story of how our tiniest biological blueprints dictate the entire grand architecture of human life.
Well, to kick things off, look at this stark contrast. It really grounds our whole journey today. When we look at infant mortality globally, the data tells two very different stories. Over in developing nations, an overwhelming 95% of infant mortality is driven by environmental factors, things like poor sanitation or infectious diseases. But then look at Western countries. Half of these tragedies in the West are locked directly in our genetics. 50% of infant mortality is due to genetic birth defects. So as we continue to solve global environmental challenges, this microscopic genetic frontier really becomes the most critical landscape in pediatric medicine.
To make sense of all this, we'll break our journey down into 5 stops: errors in fetal development, the chromosomal level, single gene mutations, cellular storage overload, and finally, the stages of childhood. Let's get right into it. Section 1, errors in fetal development, or when morphogenesis goes astray. So starting right in the womb, let's talk about teratogens. What is a teratogen? Basically, it's any agent, a chemical, a drug, or an infection that induces developmental defects during fetal life. But here's the catch. The human body isn't equally vulnerable at all times. The impact of a teratogen heavily depends on individual susceptibility, the dose, and most importantly, the timing. Timing is literally everything here. Most of these critical errors happen very early, specifically during the first trimester, right when the foundational structures of the fetus are rapidly forming. And when morphogenesis goes astray during that critical window, pathologists use some very precise terms for the structural anomalies that result. You have agenesis, which means the complete absence of an organ, no trace of it. Then there's aplasia, where an organ completely fails to develop, leaving only a tiny rudiment behind. Hypoplasia means incomplete development, so the organ is there, but it's undersized. And finally, atresia, which is the incomplete formation of a lumen or the opening in hollow organs. We actually see these kinds of dramatic, rapid-fire structural failures in devastating conditions, like the anencephaly spina bifida complex. You've probably heard of some notorious historical examples of teratogens, right? Like the torch complex, which is a group of maternal infections that can cross the placenta. Or thalidomide, the mid-20th century drug that tragically caused severe limb malformations. It's a heavy topic, for sure. But the crucial takeaway is that by studying exactly how these teratogens disrupted development, medical science has learned volumes about the precise, delicate, and frankly miraculous sequence of events required to build a healthy human being. Moving to Section 2, the chromosomal level, the colorful instruction manual. Let's zoom in closer now, past the organs, straight into the nucleus of the cell. And here we find the magic number, 46. That is our standard, brightly colored 46-volume instruction manual. Inside a normal human cell, we have 44 autosomes and 2 sex chromosomes. This incredible microscopic library holds literally all the data needed to construct and operate you. Cytogenetics even looks at fascinating little markers like bar bodies, bent structures in the nuclei of female cells used for nuclear sexing. It's an incredibly organized, vibrant world of genetic data. But what happens if that library gets disorganized? Well, we can see a few different types of errors. We classify chromosome shapes based on where their center, the centromere, sits, like metacentric, submetacentric, or acrocentric. Sometimes we get structural errors, changing those shapes entirely through things like translocations, where pieces swap places, or deletions and inversions. And then we have numerical errors. These are often caused by a process called non-disjunction. That simply means that during cell division, the chromosomes just fail to separate properly. So you end up with one cell holding too many volumes of the instruction manual and another with too few. And these numerical errors manifest into very distinct physical realities. Take Down syndrome, for instance, caused by trisomy 21. That means there are 3 copies of the 21st chromosome instead of the usual 2, making it the most common genetic cause of mental retardation. We also see abnormalities in the sex chromosomes. Klinefelter syndrome presents as 47XXY, typically involving testicular dysgenesis. Conversely, Turner syndrome is marked by 45X0, meaning a whole X chromosome is just missing, resulting in patients presenting with short stature and a characteristic webbed neck. It is wild to think that a single missing or extra chromosome can completely alter the trajectory of human development.
Section 3, single gene mutations, errors in the DNA strand.
Okay, zooming in even further from entire chromosomes, we arrive right at the DNA strand itself. So what is a mutation exactly? Simply put, it's a permanent change in your DNA. It could be a point mutation where just one tiny nucleotide base gets swapped out, or it could be a frame shift mutation, which basically shifts the entire reading frame of the genetic sequence. These microscopic typos in the code we inherit from our parents completely rewrite the cellular instructions, having massive cascading effects on how the body functions. When we're talking about single gene mutations, it's a strict game governed by Gregor Mendel's classic rules of inheritance. It breaks down into two main categories. In autosomal dominant disorders, like Marfan syndrome, you only need to inherit one faulty gene from one parent. That one faulty gene completely dominates the normal one. But in autosomal recessive disorders, like cystic fibrosis, both parents actually have to be carriers. They each have to pass down a faulty copy. If you only get one, you're a carrier, but you won't show the symptoms yourself.
But, sometimes it isn't just a simple on or off switch. Single genes rarely tell the whole story, because our environment is continuously dancing with our DNA. This brings us to multifactorial disorders. Common conditions like diabetes, hypertension, and cleft lip are the result of a really complex interplay. You might inherit a genetic composition that primes you for a disorder, but it still requires those environmental influences, like lifestyle, diet, or external triggers, to actually make the disease manifest.
Section 4, cellular storage overload. Let's look inside the lysosomes.
For our final microscopic zoom, we are diving deep inside the cell's tiny organelles to look at inborn errors of metabolism. Think of your cells like bustling little cities. They constantly generate waste and metabolites that need to be recycled. But when a genetic defect stops the body from producing a specific enzyme, those metabolites have nowhere to go. They just start piling up inside the cell, creating what we call storage diseases. And the most common site for this disastrous pileup? Right inside the cell's internal recycling centers, the lysosomes.
Let's look at 3 notable traffic jams. Each of these traces back to one specific missing enzyme. In von Gierke's disease, the body can't break down glycogen, so it accumulates heavily, severely enlarging the liver. In Niemann-Pick disease, there's a devastating accumulation of lipids called sphingomyelin and cholesterol, which severely impacts organs and the central nervous system. And then there's Gaucher's disease, where a substance called glucocerebroside relentlessly builds up inside the body's phagocytic cells.
Now, what's really fascinating here is how this actually looks under a microscope. When pathologists look at these engorged glucocerebroside-stuffed cells in Gaucher's disease, they don't just see a sick cell. They see a really specific, vibrant texture. The cytoplasm looks exactly like crumpled tissue paper. It is this visually striking, incredibly colorful, almost artistic marker of microscopic dysfunction. It turns a devastating genetic error into a highly recognizable pattern right under the lens.
Finally, Section 5, the stages of childhood, the lifespan journey. Having explored the deepest microscopic flaws, let's zoom all the way back out to the human timeline. Understanding all these genetic foundations is absolutely vital because kids aren't just miniature adults. A child passes through 4 distinct pediatric stages: neonatal, infancy, early childhood, and late childhood. And each of these stages carries its own unique physiologic and immunologic risks. A genetic defect might manifest right away as a structural anomaly in the neonatal stage, or it might silently build up cellular waste that only becomes critically apparent during early childhood. The timeline of the disease is always intimately tied to the timeline of the growing child.
So I want to leave you with this thought to consider. If studying our tiniest microscopic flaws reveals the miraculous complexity of human development, how might decoding these biological errors eventually lead to our greatest medical triumphs? Every pterogen we identify, every chromosomal error we map, and every single gene mutation we decode gives us a clearer picture of the human blueprint. Illuminating these genetic shadows is exactly what will light the path to future cures. Thanks so much for joining me for this explainer on genetic and pediatric diseases. Keep wondering, keep learning, and I'll see you next time.